to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 83rd episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO servicing the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Guarino, and I have been in the bioanalytical LBA and biomarker space for over 30 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins. I've been in the bioanalytical LCMS space for around 30 years also.
John and I are members of the growing scientific advisory team here at KCAS Bio. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS Bio services. We are delighted to have you listening to the 83rd episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we have from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. Today's a little bit different in that John and I are jet sitters. We've been traveling across the Atlantic. John, you're in a hotel room, correct?
That is right, and as a result, I don't have my usual microphone, so I might not be as clear and lucid as I normally am, so apologies for that.
Well, regardless of these jet setters, we're fired up to have you here, and we are looking to a fun episode today. To kick us off for podcast number 83, John is going to go over today's podcast topic a bit before we jump into the news and resources. John?
So as usual, we've got a few items to discuss on the news front, but our main topic today is, as we've just come back from the European Bioanalysis Forum Open Symposium, thought it might be good to just have a discussion about what the conference is, how our impressions of it, and just general thoughts on the meeting.
Well, with that, John, let's just jump right into the first article. This one's near and dear. It's anything due to world health and just in general the effects that drugs can have globally. Why don't you kick us off with the first one about some malaria drugs? Yeah.
Sure. There's nothing like a good story to bring down the mood of the day.
Yeah.
So this is the headline is resistance to crucial malaria drug detected in severely ill kids in Africa. And of course, malaria is a huge problem in Africa and other places. And so it's a constant battle to try and get on top of the disease. I know there's vaccination programs ongoing, but for severe malaria, artemisinin, let me try that again. I should have practiced this morning. Artemisinin, which is a key malaria drug, and the advantage of it was like seen as a gold standard in terms of malaria treatment, particularly for children with severe disease. So again, talking the scale of malaria in Africa, 95% of all deaths from malaria globally occur in Africa, and children are the most badly affected population. So artemisinin has been a treatment of choice, but this study actually started to detect resistance to the bacteria in children with severe malaria. So they'd seen resistance in just malaria, but not with severe malaria, and this is where it's called into play because it reduces the treatment time and cures children. But what they've seen is that actually they're seeing the ability of the virus to mutate, and actually, obviously, then the treatment time is taking longer. In the study they did, kids were cured, but just the treatment time took a lot longer. So it's basically a warning for artemisinin and how it might be affected as a treatment option in the future.
Yeah, malaria is literally a tough bug, isn't it?
Mm-hmm.
Kind of the way it can, this is the way it's transmitted, right, John? Through the mosquito, and
yep.
As it gets in, it's got all sorts of. I think I studied malaria a little bit as an undergrad. That was back in the 90s, John. That seems like a whole nother generation ago. But the life cycle of it within the body has its own kind of states where drugs can't get to it. So this is a little bit of a signal, right? They're saying, hey,
Sure.
this might be building some resistance. But what as I'm going to quote GI Joe, if you know who that is, knowing is half the battle, if you remember that kind
Sure.
of saying. But this is it's an interesting read, and certainly didn't realize it's about 95% of the deaths globally from
Yeah.
malaria are in Africa. Big numbers there. You want to tell a little bit more about the study itself, John? It was some
I think
interesting numbers.
I'd just pick up on one piece. We may touch on other things, but I think it's interesting that there's basically there has been partial resistance, and in this study they looked at the genome of the parasite infecting children, found that 10 participants had one of two types of mutations. One mutation basically led to artemisinin taking longer than usual to clear the parasite, but for the second group, it cured and then there's a recurrence. So they basically, their thoughts were that the recurrence might have been caused by resistance to lumefantrine, a partner drug, in the treatment regimen, but more studies are needed to evaluate that. So, it's just, I think it's just interesting that it tells you how tricky these diseases are to deal with. That you think you have a gold standard, but nature's dodgy. I mean, it's tricky, and so you have these mutations, and it basically means that research can't stop. You might think you have it, but you don't, because nature will find a way around it, and then you're back to square one. So there's obviously still research needed in that area.
Yeah, it's it's Darwinism, right? These are evolving.
Yeah, absolutely. Yeah,
and it's just such a great study if you're any of the young scientists that might be listening. This is certainly, I feel like this is where I kind of cut my teeth, is in like studying pandemics and epidemiology and just the whole life cycle of this, because it's such a tangible thing. It's happening. This is within our lifetime, right, John? You think of evolution over maybe millions of years or thousands of years. No, this is like they're seeing this in within our lifetime. And the bug, the parasite, this falciparum is so falciparum, right? That's the bug. It's not only does it have like 5 different stages within its maturation process, from the mosquito to how it goes throughout your tissue, your circulation, and how it aggresses or grows, right? How it grows. Now it's mutating on you, John, right? So now it's just a powerful thing. It's so symbiotic. I think that's just to me, it's a fascinating study, and certainly something that I think helps you as a scientist if you can understand these types of I can think of them as like evolutionary pressures, right? So we're given the immune system of the individual along with the antibiotic puts a lot of pressure on the bug to mutate, and hence that's why it's mutated. Just absolutely fascinating. But this is why we like medicine, John. Hopefully these doctors can continue to make the next generation of antibiotics to help.
I mean, you say in our lifetime, in terms of, I was just looking at this as you were talking, artemisinin, and there are a bunch of derivatives, but it was first discovered in 1972. So I mean, totally in our lifetime. And actually, it was discovered by two UU who shared the 2015 Nobel Prize in Physiology or Medicine for discovery of artemisinin-based combination therapies. Yeah, there you go.
Yeah, no, it is. It's John. When people think about evolution, they don't think it's gonna take
decades.
Yeah,
exactly. But it's like say it's a constant battle. So let's move on to the next one, because this is well right in your ballpark. But
sure.
again, this is we talked. So this is basically. Again, we don't usually focus on single companies, but this was a news article that was too good to pass up, and it's basically Bristol Myers Squibb has linked its C19 directed CAR T cell therapy to sustain clinical responses in people with systemic lupus erythematosus (SLE), showing that these kind of therapies could have actually potential for resetting the immune system. So, I mean, obviously, CAR Ts have been around for a while, in the recent history, but really oncology focused. But there has been this discussion recently about where they might be applicable in terms of autoimmune diseases, because there's a number of diseases that if they can help you, that could really substantially change people's lives. So it's the interest in CAR T for SLE started in 2022. Some German researchers looked at a small study of 5 people, and actually up to 8 people, where they actually had people in remission from lupus, so it's basically, and in that study, those patients had a severe progressive disease that was resistant to standard treatment. So it's like I say, it's if this really has legs, this could be huge for people with these kind of conditions.
Yeah, John, I still think CAR T cell, you think a little bit more on the immuno-oncology side. We've talked about that a little bit here on the podcast, how a lot of these gene therapies, even cell-based, this is a cell-based gene therapy, right? They are moving into these more autoimmune disease, maybe more in the rare disease, kind of inflammation, certainly allergy and inflammation, or allergy and immunology diseases. Lupus is such a tricky one. It's fascinating what this did, though. It targeted CD19, which typically these types of therapeutics are for like a leukemia lymphoma type thing. But really, the therapy itself, I don't think they fully understand how this works, John. Right?
Wouldn't surprise me. It's early days yet. And if I just
sure, go ahead. It's fascinating.
Yeah. So it's basically this set is 11 patients with SLE, as well as 3 with systemic sclerosis and 1 with idiopathic inflammatory mitosis. They observed complete peripheral B cell depletion in all evaluable SLE patients after administration of the BMS therapy. B cells began to return in people with 3 months of follow-up, but were mainly naive cells with very few memory B cells, C11, C plus double negative B cells or plasmablasts. So the B cell depletion helps reset the immune system, reduces patients' ongoing systems, and hopefully frees them from the need to keep taking medicines to manage their conditions. So the BMS actually reported clinical responses in 7 SLE patients with at least 1 month of follow-up. So that's like the summary of the study, and that's what sort of draws you in terms of this. This really sounds like there's some potential here.
Yeah, and this depletion of CD19s and resetting, I'm trying to think of mechanistically what's they're just repopulating and it's coming back, and maybe over time the let's call them the diseased B cells, right? There's something in here. I think all these different forms of lupus and idiopathic diseases, these kind of autoimmune diseases, they're like snowflakes, John. No 2 patients are kind of alike, unfortunately. At least that's kind of how I was taught it and sort of think of it. And so how this sort of reboot of CD19s causes whatever sort of bad cells. I don't know if they qualify as malignant. That might not be the word. That's a technical word, John. I learned it. Bad cells. I was taught that. No, but these infectious cells, however you want to think of it, causative cells are now kind of gone. And it shows some great efficacy. I'm more familiar with an ECOG status. That's what they use for oncology patients. I'm not familiar with this SLEDAI measure, but all these patients improve remarkably and up to 11 months. And now they're talking about maybe the need for another injection and so forth. But it is fascinating that this worked. And part of me is like, I don't know, maybe I should reboot my B cells, John.
Yeah, but there is one paragraph here that says the one outstanding question is how many autoimmune patients will be willing to undergo CAR T therapy, and partly this will come down to the safety profile of the candidates. And there are a number of candidates out there. We're just focusing on BMS this time. There's also Kyverna have a drug in development, Bio and Novartis. But basically, BMS saw grade 3 or grade 3 or 4 treatment emergent adverse events in 11 of the 15 patients in the safety data set. Neutropenia, meaning low levels of a type of white blood cell, was the most common grade 3 or 4 adverse event. So yeah, it's again, it becomes that benefit versus, what's the benefit versus the side effect? Is it enough for you as a patient to risk that side effect to move forward? So we'll see where it goes.
Yeah, no, I kind of caught up to you there with the emergent treatment there, although neutropenia, it's treatable with Neupogen, right? Those types of drugs. But I kind of jokingly say I'd like to reboot my CD19 cells. I could just think about what your physician would look like if you said you wanted a CAR T cell therapy proactively. That first is a bill to it. I don't know. I'm just laughing. I'd like to see the face of the doctor where you're like, "Hey, can I get a CD19 CAR T cell?" No, okay. I don't have anything to add, John. It's just great to see, somebody who did autologous T cells again in the 90s, decades ago now, to see all these drugs kind of or all these therapeutics making their way into to treat all sorts of different diseases is fabulous. Of course, this treatment emergent thing, I think that's what, no good deed goes unpunished. You find these very powerful therapeutics. They almost inevitably come with some form of side effect, but even that over time they'll engineer it to diminish this as they continue to study it. CAR T cells are here to stay. I know this. There's still a couple. I don't know if haters is the right word, but there seems to be a few out there that are skeptical about the side effects and so forth. But I'm pretty sure if you look at the numbers, even though this had a high, again, it's hard to tell what an adverse event is when you deplete someone of their B cells and naturally they're low on neutrophils. That I don't know. But what I mean by that is there's upwards of, I think the last stats I saw, there's probably 60,000 people treated with CAR T. That this was some time ago, and it was less than 1% to 2% with the
Sure. Yeah.
So it's great, and yeah, we can move on to the next one. But I can only think of what it'll look like in another 30 years and say 60 years in terms of what these therapies would be. Maybe you can go to a vending machine and treat yourself with cancer, some allogenic.
Seriously. No matter how optimistic I am, I won't be around in 60 years to find out.
Yeah, I was thinking about our kids and grandchildren, John, not you and I. I won't be with you, man. I don't know. Who knows if I keep my body by chemistry and keep doing.
If you keep going for all these proactive treatments, you never know. It's like
I was jokingly telling someone, pull my bone marrow out and culture it in my garage and treat myself with my own T cells.
You've got a lot of possible fascinating Instagram stories you could be posting on this stuff. Yeah, there you go.
Next to my big green egg, I'm going to put a biological safety cabinet, John, in an incubator. No, I'm just kidding. Let's move on. I'm on. Good job.
Okay, next up is Atlas of Tumors reveals in 3D how cells change into cancer. And this is sort of really trying to look at what's going on. So I'm going to read the introduction because this is one where I'm sort of still trying to get my head a little bit around it. The story of cancer has long started with a single mutated cell that divides and eventually grows into tumor causing disease and death. But in this new research, basically there was a lot of effort to map in 3 dimensions how abnormal cells become malignant, and adds more evidence that for at least one form of disease, colorectal cancer, tumours can arise from multiple cells, not one. And understanding the way abnormal cells turn into cancer could help lead to new, earlier intervention methods against colorectal cancer, which has been on the rise in people under the age of 50. This is part of the what's known as the Tumor Atlas Network. It's a research consortium funded by the National Cancer Institute and launched in 2018. And so they've published a number of studies on a number of cancers, including colorectal, breast, and other tumors. And so basically, these detailed 3-dimensional maps allow researchers to see where different cells are and also how they all interact. So it's like they knew tumors were heterogeneous, which helped them to understand cancer relapse. But this was just by inferring it from other data. Now they can actually observe what's happening, and it's all down to instrumentation, like spatial biology tools for mapping gene and protein expression from tissue, as well as single cell sequencers. And so it's looking at how tumors change over time, moving from normal cells to abnormal precancerous cells into cancer cells, and it's just getting a handle on all that.
Yeah, John, I think they're pretty vague. They say powerful instruments, right? It's hard to tell exactly what they're doing here. At least I couldn't. To me, are they excising the tumor and then trying to study it, or is it still within the patient? I
I
can't
think
quite.
it's much. Actually, I'm not sure on that one.
Yeah, that it was difficult to tell, right? But that's a
I think it's looking at cells in bulk. Oh no, sorry. It's looking at cells, and it's actually looking at single cells. Is where this is, because originally the work was done by looking at cells in bulk, but this is really looking at a cell at a time with these instrumentation platforms that you have.
So it must be external. It's all relapse patients, right? So as they start to see it in that immature state, they must be removing it. In the past, they kind of talk about blendering the tumor, putting the tumor into a blender. That's traditionally like what I've researched. Someone gives me a tumor, we kind of blend it up is one way to put it, but you could look for tumor infiltrating lymphocytes, right? You can look for all sorts of. It's such a powerful, to me, immunological starting block because that holds some of the keys to potentially treating the tumor, right? That's where novel tumor antigens would exist, and so I've studied it that way. To be looking at it, so I'm guessing it's external, right? This must be
Yes.
trying to look at that growing, but I mean, John, this is so powerful. If we can start to unlock how, even if it's just colorectal or this pancreas, any of these tumors, kind of what is causing them to kind of literally mutate in front of our very eyes. I mean, man, that makes it a lot. You get a lot more targets to treat and therapeutics to treat versus most of the time we're at a solid tumor mass, and that thing's done so many things to evade the immune system, right? That the tumor itself has within a tumor, the external cells are like decoy cells. They're out there just shedding themselves, almost kamikaze in the immune system is how I think about that, where the core of it is where it's kind of protected and it just sloughs off and grows and sucks up all the nutrients in the surrounding area and just takes over. And so then the tumor microenvironment itself, because of the way the tumor's kind of evolving, it just takes the immune system and either kills the immune cells in a variety of ways, or it just turns it from acute to chronic, right? So if it's producing TNF alpha and IFN gamma and killing the tumor and you're okay, if that tumor starts to get those cells to go from producing those cytokines to more what they call like type 2 ones, like IL-4 and IL-5 and things like that, then the tumor can grow. It can actually nurture some of the antigen presenting cells into a different state. So then therefore the acute response and the killing of the tumor goes away. So, how and what this leads to is probably the sky's the limit, but it's just great. It touches almost every single famous university in the country as well. This is a fascinating story, John.
And I think that's in line with what one of the contributors said, that actually someone was asked about this and saying that you'll see down the road, 10 years down the road, so you still might be around to see, or should be around to see that, hopefully, that people will be citing both these projects in terms of the data that this has thrown up that's really helped, then help with development of drugs down the line.
I mean, John, they're probably discovering all sorts of new, I think of them as tumor, not that there's tumor associated antigens, but just the genomic information you're going to get. So much to be able to see it I don't know, it's blowing my mind a little bit, right? I mean, there's all sorts of dermal ones where, people can put some sort of treatment on say the skin, grow a tumor, and you can keep a wand, a microscope, a confocal, and just watch it grow. But this is a whole nother level of single cell.
Yeah, sure.
Usually you have to put some sort of dye to do that, fluorescent dye on it and watch it and so forth. But this is just, I don't know, it's the top of my head just went, and then I was thinking about, wow, what does this is opening up a whole new line of like research and therapeutics, and then you cited it. 10 years from now, people will be citing these projects. And undoubtedly, again, if I'm a young scientist listening, I'd move towards this. This is a great area to research. Yeah,
Yeah,
fantastic.
and I think it's interesting because this is what I think where I seized on it. Where I was looking at this earlier. It's like prevailing theory is that cancer typically starts from a single ancestral cell that acquires a mutation. So then you get division, more cells, offspring get mutate from there, and so it sort of evolves from there. But a number of these papers looking at colorectal cancer suggest that some colorectal precancers may start from several cells as opposed to one. And so that you're then chipping away at your prevailing norms. So that's what the kind of thing that this is is leading to in terms of conclusions people are getting. So yeah, you're right. So then your treatment becomes different as to how you may address this. So yeah, I just thought it's very, again, another nice, interesting complementary.
Yeah, maybe one last point for me here. Like a lot of head and neck tumors, cervical cancers, anal, penile cancers, all of that are virally infected, John. And the etiology of it is definitely HPV.
Mhm.
I don't know if you've probably heard me I have this belief, and I'm not alone, that every I believe every kind of tumor cancer has some sort of viral component to it. Now, it might not be the true cause, but it's opportunistic. And I think if we had the right tools, and maybe this could prove some of my theories, I wonder how much they're going to find. Maybe these are virally infected as they're what's causing that reoccurrence. Again, I don't know, but I would be fascinated to see if anybody's looking into that, just trying to see if there's some sort of viral component. But I'm a firm believer in viruses being either the cause or exacerbating or synergizing with certain tumors, or any tumor. It's just tumors rapidly grow, viruses rapidly grow. It's like a fly to the light, John. That's how I think of it. So we'll
Sure.
see.
Yeah,
But
I mean,
anyway, it's all. But maybe we can move on. I know we're running. That was a little bit long of an intro. And we got some investment stuff I really would
I
like
think
to talk about.
this is actually really, yeah, this is timely, and it's the reason why I'm in a hotel room doing a recording rather than comfortable in my room at home. Because I think we wanted to be talking about things like this before all the actual decisions made. But several months back, Novo Nordisk um, signed an agreement to purchase Catalent, and Catalent being a CDMO doing contract manufacture of some of these difficult to synthesize drugs. And this, yet again, we're back in GLP-1, but both Novo Nordisk and Lilly have gone through phases of having shortages of drug supply. And like, because the chemistry is complex, it takes time to make these things, so they need third parties to help. And both have gone at this different ways to try and relieve their supply. But so Novo Nordisk, one of their strategies, as well as it expanding internal capacity, was to buy Catalent. So it meant that they had a capable supplier on hand to help with their drug supply issues. But we're seeing more that there's now the sort of backlash or concerns are being raised by other individuals in the field, because, as we said when this was first announced, this Catalent are an important supplier for other drug companies. So if they're now owned by Novo Nordisk, how does that then affect the supply for these other companies? And both Catalent and Novo Nordisk have been very sort of, they've said, well, of course it's not going to affect things, but as an outsider, it's very difficult to see how it wouldn't. So what we've seen, and there's a number of news articles that we might just touch on here, but there's so basically, consumer and union groups call on FTC to challenge the Novo Catalent deal. So they basically, so there's around 10 consumer groups and 2 labour unions wrote to FTC's chair, Lena Khan, warning the deal will affect competition for weight loss, cell and gene therapy drugs. We believe this transaction, if consummated, would be harmful to competition across various therapeutic areas and ultimately reduce patients' access to these vital treatments. The letter was, I think it was either late October, early November. And the reason why this is becoming timely, there's like the Roche's chief executive said, although they're not really affected by it because they have other strategies, they still see that this could hit smaller drug companies. And in that case, it could hit Roche because, as we know, a lot of these big pharmas are often going on buying sprees, and they're always looking for small companies with good ideas that they can hopefully partner or buy and take those drugs to market and make money on them. So that there's obviously big things at play here. And then the last, I mean, we've had Catalent trying to reassure customers that this won't happen. But the reason why this is timely is the European Commission are actually looking to make a decision on this deal on December 6th. At the meantime, they've sent out questionnaires to customers, competitors and suppliers as part of their investigation to try and compile feedback. And so this is all this is a fascinating scenario here, because it's one of those things. It was when you first heard about the deal, you thought, oh, well, why? And then you thought, then it's like, well, that makes perfect sense for Novo Nordisk. But then the ramifications could be very, very interesting, and we'll see where it goes.
Yeah, my wife works for Catalent. I'm hesitant to want to comment too much on this, but I just think the deal will go through. Some of these things you're talking about have been, they're kind of rising to the surface here, but I think in general, people have found alternatives, and but I guess we'll find out, John. But I don't have too much to add. This is somewhat near and dear to me.
Sure.
Because of the my wife haven't worked there, so I'm pretty familiar with all this. And but just as the public reads here, this is a December 6th setting, and soon thereafter, this should be behind. But this has been going on a long time, John. This is probably 18 months or something. I think it's been quite some time.
It only said deal in February, and it was announced in February. It's the European Commission says December 6th for decision, and then actually the Federal Trade Commission at the end of the year. I think, like I said, to me, it's interesting, and I know the Catalent link for you, so that's fine. But I think if this goes through, what will be then the ramifications? Not even in terms of the Catalent customers, etc., but will other companies follow this model, i.e. other pharma looking to buy these types of organizations, and will that actually constrict the market in terms of available
CDMOs? Yeah, it's a slippery slope, right? Yeah,
I think that's really more where the interest comes from me. It's like, this is quite an unusual deal on the surface. So what if this goes through? What might come of it? So we'll see. I wanted to raise it because, like I say, in terms of how it might affect the industry, this is quite a significant deal, I think.
Yeah, it is one where it's a head scratcher. If Novo is allowed to do this, then what's preventing any other pharma to go out and just buy a CDMO like they did? And this is not a small organization.
No, not at all.
It's over 50 sites or something. I mean, it is big. So it is just even when it happened, it made you scratch your head and think, well, they shouldn't allow that. But here we are, and my understanding is I don't think anything's blocking it. In that, I think what came to is some sort of agreement that all the companies that were working with Catalent and find this to be a threat are now just moving on. But it does seem to be a little bit of a monopoly, I guess is the word that comes to mind, John, in that if I'm a biotech and I'm Novo and they're sitting here seeing all this in the CDMO space, why wouldn't I? Doesn't that give you a little bit of insight? So I don't know. It's just this. Yeah. I'm just glad I'm a scientist, John. I don't want to think about this too much, but it is definitely something that is potentially a bellwether of how the advancement of contract. This is how contract research, specifically in the CDMO space, may be going. But
yeah.
I guess I don't know. Novo has all this GLP-1 money now, right? They've got all that. So some of these other organizations, I'd be looking at this going, well, what am I doing? And then I think to that point, some of them have opened up manufacturing facilities, just not CROs, right? So I
Yeah,
don't know.
yeah. Well, I'd like to say it's interesting. December 6th will be obviously we'll see what comes of that and
Yeah.
it goes from there. Like I said, it was too good to pass up without
No,
passing
it's
on it.
juicy. It is
So
super juicy, and I have my thoughts on it. I mean, my wife's been there a long, long time, but it's just there's still. Well, let's move on. Yeah.
yeah, absolutely, it's absolutely fine. Yeah, yeah, not a problem at all. So let's move on to the main topic. We try and keep the fairly regular recordings of the podcast going, but our travel recently, particularly recently, has made things a little bit more difficult in terms of trying to find the time where we're all here. And it's not just Dominic and I; it's also Jeremy who's been gallivanting all over the globe doing other things. So both Dominic and I were at the European Bioanalysis Forum Open Symposium last week in Barcelona, and we thought it might just be good to have a little discussion about how all that went and just impressions and what we thought of the meeting, etc., etc.
Yeah, John, it's my first one, right? And the Europe, for those that aren't familiar with the European Bioanalysis Forum, it, John, it was one of the better meetings I've ever been to. Certainly, and unpacking what I just said there, what makes for a good meeting is one that has, I'd say, not too big, right? I think that's an important first step. Sometimes you go to a meeting where there's over a couple thousand people at it, and you're trying to go to different tracks, and it just gets a little mucky.
Mm-hmm.
This only apparently they had just expanded it, right? They had gone from 400 to about 600 attendees. The meeting itself started 16 years ago now, was it, John, or 15? I feel like.
I think we were at number 17, but there was also virtual ones during COVID. So I'm not sure how they counted. I
And
think
and only like 10 years ago did they let CROs in, right? And they still
no,
don't let any regulators in.
no. It's been there's so there's let me backtrack.
Sure, sure.
So the European Bioanalysis Forum, it's a little bit confusing because European Bioanalysis Forum is actually an organisation. Everyone thinks of the meeting in November as the European Bioanalysis Forum, but that's not what it is. That's actually their open symposium. They do a lot of workshops through the year, and the actual forum, it was initially pulling together pharma companies from all over Europe to really talk about bioanalysis, regulations, things that they were doing that didn't make sense, that they could possibly lead on. They're always looking for initiatives where they can provide input as to maybe this is the direction we should go on, such as things like fit for purpose validation for biomarkers. They're very big in trying to reduce your footprint for consumables, things like that. So the Open Symposium in November is that main meeting, and CROs have always been there to present, allowed to present. I think where the involvement of CROs increased is in the actual European Bioanalysis Forum. So they become member companies who are there to discuss where the European Bioanalysis Forum as a group wants to go, and it's based on member companies' feedback. So you'll see a number of talks where they've done surveys with member companies and give feedback on various things. I mean, I was in one, and they just they were still discussing setting mid-level QCs
Oh yeah.
for LCMS, and I'm thinking, why are we talking about this? And to be fair, that's not Europe. The EBF, I'll just refer to them as EBF. They want to get away from those kind of trivial questions. They want to be talking bigger picture and getting away from really going. I mean, there was still people in the audience who really want to be told how to do things, and I'm sitting there thinking, but we have such a wide range of assays that ultimately to have a prescripted here's the acceptance criteria, here's how this should be, or here's how this should be. It doesn't fit everything. So you need some fluidity there. And so I don't want to have everything prescribed to me. That it's like you have people who will stand up and say, I want this language really tied down firmly. And it's like, well, why do you want that straitjacket? Why don't you want to have that wiggle room, knowing that, for 95% of your assays, they're going to fit a model, but there's going to be that 5% you want that wiggle room to be able to deal things with slightly differently. And I think EBF are big on that kind of thing. And like I say, I like what they do as an organization. To go back to what you say about being a good meeting, it's been in the same venue for, I think, the first two were in a different hotel in Barcelona, and for all the rest since, it's been in this one hotel, which used to be called Hesperia Tower, and now it's the Grand Tower Hyatt or something. But you're not in Barcelona. Well, you're on the outskirts of Barcelona, so it's nice in that you have access to the amenities of Barcelona in the evening, but when it comes to the daytime, everyone is in the meeting space. The talks are very scientifically focused. They've got more vigilant about stamping out noticeably commercial slides, and I'm sure you've got something to say on that.
Oh yeah. No, I think that is what, I appreciate that tonality. I don't know if you were done yet, John. It's
No,
okay.
I'll finish up very quickly. So there's very little commercial, which I have to say I really welcome, because I've sat through some talks that have been just awful in terms of the person starts, you see the topic, you think this could be really interesting, and then it's a sales pitch, and it's like, yeah, really, that was 20 minutes of my life I'll never have back. To go back to the regulator piece, they did. It was said at the beginning why they don't invite regulators to speak at EBF. They're welcome to attend, but what they found is that when regulators stand and talk at a meeting, it tends to be they have to. It's obviously a part of the condition of their employment. They have to state that this is their personal impression, personal opinion only, not that of the organization they're representing. And then when you actually look at their talk, they don't really say very much. So because their hands are tied, I totally get where they're coming from. So that, but EBF want to lead. They want opinions. They want people to be throwing things out there for discussion. So that's why, for them, the regulator presentations aren't necessarily great in that particular forum. The other piece of EBF is not only do you have the talks that you're all in this one venue, but the trade show is also in one room, and all like social events are in that one room. So there's a lot of mixing between vendors and customers, and that is a really good thing too. So there's a lot of reasons why this is a really good meeting to attend, and it's why it's one I've tried to attend as many times as I can. I think I've done it for all but 3 or 4, and I've been really lucky that I've been able to do that.
Yeah. You make a good point about what makes a great meeting, right? And one of it is the venue, and the venue's perfect. There's as you indicated, no one really left for lunch. Part of it was there was only maybe one place that was really walking distance that you could sit and eat at. But more importantly, they had good quality food there, and it encouraged you to engage everybody over lunchtime. The meeting was big enough that, like, not by the end of the first day you had met everybody either. So it was like the right size, right? So like each time you had a coffee break or a lunch, and they were clever. They only had, instead of like 2 coffee breaks, they really only had 1, because things went late there, right? We finished up at like 6:30. And so the combination of that just gave a great like flow to the meeting, where it didn't feel like you were just staring at somebody come the second day coffee break, right? So that helps out. And then, as you mentioned, it is so scientifically centric, John, right? Like that was the powerful piece. There was, everybody who was there has almost been vetted, right? They just didn't let in, even though there was, a bunch of booths and everything. You and I are PhD scientists, John. We're not, so the people who are attending this are really highly scientific. And if you didn't have good scientific chops, you were kind of getting left behind. And so that made it really powerful. And then even the whole process, you kind of touched on it not being salesy. I'll tell a quick story about the presentation I gave where submitted the slides on time, by the way. I'm happy about that. And then they came back from the head of the organization, and he's a real stickler, and I kind of respect that he is. Phillips' his name there, and so he, they came back and said, "Hey, we love your scientific content, but unfortunately, we're going to reject your slides because it's too salesy." And I thought, what are they talking about? So on about 3 of the slides in the case studies I gave, there was 5 case studies, and on a couple of them it said, "We at KCAS Bio solved the problem by," and they didn't like that. It just said, "Hey, your audience knows you solved the problem," and just I had to literally reword it to "the problem was solved by doing this," and that's the level of kind of meticulousness they have about it not being salesy in any way, shape, or form. And John, I kind of respect that. I'm not trying to make fun of that.
No, no,
But I
no.
want to be clear about that. I'm like, hey, I'm like, that's a good approach, so that you're just not having like, to some degree, I'm not going to, WRIB is still a great meeting, but occasionally someone will slip through it, like you said, you're like, wow, this title is nowhere near the presentation I'm watching, and I just lost 20 minutes of my life watching this, infomercial. But wait, there's more, right? People kind of sound like that sometimes, like it's an OxyContin commercial or something. But so that you're right, the venue, I think it's steeped in tradition. It felt like that was kind of cool. And it wasn't, sometimes these meetings can become intimidating, John. And what I mean by that is it's like these subcommittees all know each other, and I kind of think of them as like a Jedi council, right? Like, oh my God, who are these? But they were really warm and engaging. Anybody that I spoke to welcomed the opportunity for KCAS Bio to become more of a participant, because every session started with the survey. That's a big part of it. That drives the content of the meeting. So that's really, when you listen to your audience, that's why the meeting's so successful,
Sure.
right?
Yeah.
That's what happened. And I sat in on the biggest talks were on singlet, right? There's a big movement to remove in the ligand binding assay space, as well as in like the PCR space. And can't speak to molecular to mass spec, but they want to go away from duplicate replicates to singlets. And there's now enough evidence and data to do this. It's just, I don't know. I'm still a little bit torn by this, and I guess
Sure.
so long as the method allows for it, you can do it. But I think your a priori criteria around repeating a sample becomes more challenging, especially if it's just through grub test or stats. You can just tell it's an outlier. What do you do with that? Do I have to repeat it, right? Where if I've got 2 data points there, I can make some other decisions, but I probably still got to repeat it. But that was a fascinating topic, John, and that is the audience. It wasn't anything I thought about, but you talked about it. People think it can be a huge cost savings. I don't know necessarily about that, because you still got to manage the data piece. That's really where the people who, if you haven't outsourced, or if you haven't worked at a CRO, it's not whether I do duplicate or singlets, or that's all still kind of baked into sure, I get more efficient with maybe the number of samples I can turn, but managing that data piece still has the same sort of QC and QA to it, right? But
Sure.
that was one talk, and then there was heavy on PCR and some of the supportive cell and gene therapies and some of the challenges they have there, and then I sat it in on hybrid. But all of them started with surveys. It was great, John. Maybe I'll let you back in, but it was the way the functionality of the meeting, the core values of the meeting. Sometimes people advertise that way, and they don't. Their words and actions don't always match up. Here it did. It was impressive how to the T they were, and they didn't just say, "Oh, do this," and then go over here and behave a different way. Everybody was kind of in that mode of, "Hey, we're here to learn. We're here to," like you said, it was provocative at times. There was you could go up there and say something that was like heresy in the bioanalytical world, like for instance doing singlets. That seems crazy to me, but that's now becoming a real thing, and I wouldn't be surprised if you're going to see us adapt to that. So it's really, this is how change happens. It has to be somebody. There is, and I'll end with this. There's no next level, John. This is it. There's no other meeting that's more powerful in Europe than that meeting. It's this one. It's the who. It's like the Super Bowl is what I call it, of meetings, and it was wonderful, John. I can't believe. I don't know what I was expecting because of the the hectic travel I had, right? I did a panel while I was there. I had to do a presentation. So I kind of looked at the content and thought it was interesting, but I didn't really think about how and what it was going to be like because I was just so entrenched in what I had to achieve. But man, it was really good, really informative, really warm and friendly. We kind of all waved the white flag in order to kind of move science forward. I felt like we were moving science forward, and it felt like it was a great investment in both time and money for us to attend, John.
Yeah, I totally agree. Yeah, like I say, I've been a strong advocate because I've been lucky enough to do it for years. When you say about the size of it being right and being able to meet everybody, I didn't meet everyone I wanted to meet. I still managed to miss people who beforehand I said, let's get together. And I passed like couple of people, and I saw them in pass and said, I will catch you later. And they said, yeah, I'll catch you later. And we still didn't manage to link up. And it's like, why did we do that? But it's because you were so busy, because it is an intense meeting. Yeah, I mean, it's really only 2.5 days, but it's an intense 2.5 days because it's long days talking and learning. And the ADC sessions were great. The microsampling sessions were great. And I have some thoughts about the microsampling piece I want to talk about to you separately. I sadly I missed the Legos because I forgot to set my alarm clock, and more that's on me. But it was. I think actually this was a really good one in terms of the standard of presentations. I think this year are as good as they've been.
Let me interview you a little bit, John. Let me ask you a couple more just a couple things before you dive into some of the.
No, I wasn't going to say much more than that. I wanted to acknowledge and kudos to all the presenters that the standard of presentations was very high.
Well, yeah, that they said they had to actually expand it to, I think, maximum. The building, it seemed like the largest auditorium, couldn't fit more than 600.
Yeah.
I don't think the meeting's going to grow.
I think that's why they stick to that venue, so that there is a cap there. And they said they actually rejected the number of.
Yeah, that's what I was going to say. They had a record number of submissions for posters and presentations, and they did reject a record number as well. That's why I thought it was kind of cool. We had 2 posters and 2 presentations, John. So we kind of hit the mark. That was good.
Sure.
Everything we submitted, our Lyon colleagues had 2 posters there. Fabian and Musin both had them, and then you and I presented. So it was really good. But John, how many of these have you attended, right? So maybe we got to remind people that you spent a lot of time. You are obviously European, right? So you've spent, how many of these have you been to? I know you've been to like 4. Is this your 4th as a KCAS Bio?
No. As KCAS, it's, I think it may be my 4th, but Advian / and their subsequent iterations, I was there. I was there for the first one. Maybe I think I maybe missed 3 or 4.
Yeah,
it's not many because it goes. My role in my previous job had a lot of similarities to this, but I was running an operation, but I also did a lot of involvement with business development because we didn't really have a very evolved business development team. It was small, so they didn't have the bandwidth, so they relied on people like me to step in and help. And it's not knocking the organisation, it was just different, and I was lucky I was a beneficiary of it. But we had a lot of customers in Europe, so EBF was an ideal meeting to attend, or EBF Open Symposium was an ideal meeting for me to attend, to actually link up with some of my customers, and I would add client visits on before and after if I could as well. So it was always that building FaceTime with clients, because it's such an important part of building a relationship.
Yeah. So fast forward now, and you're attending this meeting, and you probably saw you can share. But John, I've been to as many WRIBs. I was shocked at the 15 faces that I recognized there.
Sure. Yeah.
And as you said, I could even think of a few people where I had some great conversations with some people, but then also went by and said, hey, I'll catch up with you just as you said. And then sure enough, it was like a whirlwind of places and all the tracks, and they kind of go around with their bell and shoo you into a meeting or something. So you're supposed to be at the meetings, and I wanted to be at these meetings because the talks were so good. But, to your point, and where I was kind of going is it's a who's who, John. I was pretty impressed with the number of people that I knew there. I did not expect the amount of the presence of, and maybe I just been doing this so long. I'm what did Bucksat tell me? We'll make you a rock star overnight, and it's taken like 11 years, John. Right? So, but it was a little bit flattering to just realize how many people there. And then I'll add, John, what's most impressive, and this is, I want to give a shout out to like Charisse and our Lyon business development team. I had so much fun seeing Charisse. She's such a lovely person.
Sure. Absolutely.
Smart person, such a great asset to our organization. Unbelievable how she can flex from English to German to French. She's so, I'm looking at her, oh my God. But they when I had about, sitting at our booth kind of hanging out, have at least a dozen people come up, and I'd say like, oh, have you heard of KCAS Bio? Because it's just, I'm so used to saying that because I was employee 43 all these years ago and no one really knew who we were. They all said yes, John. And then here's the shocker. A couple of them, I even was bold enough to say, oh, do you know who Dominic Guarino is? And they were like, yeah, I've listened to your podcast and we have some adventures there, don't we, John, if we have time? But that shocked me, John. So I think it was, to me, it was impressive. And we'll get back to the conference and you can share more, but it was just the level of presence that we had and the notoriety. I guess, I don't know if notoriety is the word, but I was just really impressed with how many people knew exactly who we were and what we were about. So it makes for a much easier conversation with somebody when you don't have to like, kind of say, hey, well, we're a 44 plus year organization, blah, 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 right? We've been audited by the FDA 18 times and we've got over 300 drugs on the market. You can just say, oh, you have you guys are in LBA space. What do you need to know, right? It's just a different conversation.
Absolutely. Actually,
it was really surprising. I can't think of a single person that said, no, I had not heard of you. So good job to our marketing team as well as our business development team for preparing us. But maybe back to the science.
So can I just chime in on that? Yeah. So yeah, you're absolutely right. I didn't strike me at the time, but I don't think I said there was anyone asked me about who are you, what do you do type of thing. All the conversations were already that next level on from there, and that's very different from any other meeting I've been at.
And maybe it's because the level of scientists there too, right, John? Maybe it's the level. So sometimes you go to like other meetings, even RIB here in the U.S. There's a lot of junior members. I didn't feel that way. This seemed to be, you had to be a pretty senior member of your organization to attend.
But a lot of hands-on people, so it's people who really knew the science they were talking about.
They're hands-on people, but that you could be a decade, right? You start at 22 or 23,
sure, absolutely.
And you're still only 33. That's still kind of junior for guys like you and I. But you're right. I don't want to make it sound like it was a bunch of gray hairs at this. No, I'm just saying it was everybody I met knew. Even the 30-something-year-olds have been decade in the science realm.
Yeah, absolutely, yeah.
So tell us about, John, I don't know if you want to dig in a little more or we could wrap up. We've certainly gone almost a full hour here. I don't know what else you want to talk about, but for me, it was the singlets. A lot of fascinating PCR stuff. And really, and I'll just wrap, PCR still has some pitfalls in terms of managing the extraction process is still a challenging one. And what I mean by that is, do you spike whatever your target is in and then extract it? No, you extract it, then you spike it. So there's this sort of efficiency question. They're managing that, but there were some discussions around that. And then it's just, you're trying to treat a cell-based gene therapy and it's not a PK assay, so you can't follow the regs. There's a lot of discussion about not having regs, but there are some guidelines now at least on how to do it. So that was kind of good. And then the hybrid talks were fantastic.
Yeah,
they don't call it hybrid, do they? I want to clear. They really don't like that term. It's protein mass spec. That's really what they like to say. So, but you can go ahead and chime in there. But to me, it was really good. I enjoyed it.
I think I would agree that the protein LCMS session was excellent. And I mean, I was there from the beginning all the way through to the end. That was a really good session. It was interesting to see the change that now small molecule is in one more of the when it goes into the split session. It used to be that small molecule or LCMS had the main auditorium. I was only in the main auditorium once, and that was for the early talks. After that, I was always in the satellite rooms because we've now had the flip of where LCMS has become the less attended of
Well,
the.
that's interesting, John, because I did go to the PCR one, which is in the bigger room. It was not nearly as full as the protein mass spec meeting
Oh,
was.
really? Okay.
Yeah, yeah. I maybe they wanted to shift it a little bit, but that to me, I didn't go to every talk, but the protein mass spec one had the largest audience versus. I didn't go peek in, but I don't think there were enough people. That room was almost full. I don't think there was a sharing, but anyhow,
Yeah.
it's
Yeah.
interesting. It's interesting.
Yeah. Yeah, yes, I don't think there's much more to say. I think, like I say, it was a really fun experience, a really great meeting. I'll be first on the queue to go again next year because, like I say, if I can, because it's a great meeting. And
Do we want to give a shout out to our? Well, how about the museum? Right?
yeah, actually, I
Yeah.
do want to touch on that. So, usually, so again, just talk about the structure. Basically, when it comes to the end of the day, there's usually a small cocktail session mixer, which some of us were on antibiotics and couldn't do anything but drink Coke until the last night. But but that again, it's that because it's in the where the vendors are. It's more relaxed than during the day where it's the trade show. But your vendors still tend to stick by the booths. But it still has a really nice atmosphere to it, and people will mix and everyone does talk, and you really get a chance to mingle. But and late in the evenings, it's usually when Barcelona wakes up to eat. So that's what they have the cocktail session. Then you're left to your own devices, and so a lot of us, one of the things that nice thing about this hotel is, although it's on the outskirts of Barcelona, you're probably 4 or 5 minutes walk from one of the termini of the underground system, so you can get anywhere in Barcelona, and it's a really easy system to navigate. And so if you have a restaurant, just jump on a train and go in there. That's great. But on the Thursday night, the how God names it. I'm sorry, jet lag. Jet lag got me.
SciEx, right? SciEx
It's
sponsored
it's,
it.
it's Ferran
Yeah.
who heads marketing at SciEx in Europe. He will, because he's Spanish. He or I don't know if he's Catalan or Spanish. I'm not going to distinguish that, but he will arrange an event at one of the local landmarks. And so a few years ago, I was lucky enough that I got a ticket and I got to go and do the tour of the Nou Camp, the FC Barcelona stadium, which was absolutely fascinating. And then once you do the tour, then they'll have drinks and food, and it's just a really nice evening. This year was at the Juan Miro Foundation.
Mm.
So, say again, similar arrangement. You get buses to the venue. Then you're walk through. In this case, walk through various galleries with a tour guide who will describe the
Hang on,
work,
John.
talk about
Right? Hang
Juan
on.
Miro's history. I'm
Yeah.
getting there.
Okay.
Talk
No,
about
but Ferran gave us a nice tour of the city too, right? That was
Yeah,
the.
yeah, yeah.
Yeah. Okay.
Because actually, to part of it, yeah, to get to the Miro Foundation, you're actually going through where a lot of the Olympic events were held in 1992 when Barcelona hosted the Olympics. So yeah, Ferran, well, he's very good about sort of pointing out landmarks as you're going through Barcelona. But yeah, it's a really nice event. Tickets are scarce. And so, if you can get a ticket, it's really worth getting one, because you'll see something in Barcelona that you wouldn't necessarily know about, and visit yourself, and it exposes you to really nice stuff. And again, it's a really good chance to talk to
Wonderful.
people.
Yeah,
Yeah,
you got a guided tour of the museum, right, John? We had
yeah,
music going on. They had food. You got a great. It's elevated. You could see the landscape of Barcelona. You got the tour through some of the 92 kind of was really kind of transformed Barcelona a little bit, or they built all these kind of, similar to what they did in Atlanta back in the the Olympics of 2002, I think that was, or maybe can't quite remember the year there. But similarly, if you go to Atlanta, they have Olympic Park and everything in the center of town. This is like a whole region of Barcelona they built up. They put this museum in, and John, it was fabulous. It was a good
Yeah,
time.
it was a really good tour. I mean, I'll be honest, it's the sort of stuff that artistically is way over my head. It's one where I need my wife there
Yeah.
because she she did history of art as a degree. She gets it, and she's lots of fun to do art museums with. But it's certainly somewhere where I'd need more of her thoughts
Having
on.
the tour helped. At least that
Oh
gave
yeah,
us
it did. It did. It was fascinating.
oh,
It's just like right now, I wouldn't necessarily be looking to hang a mural on my wall, but it's like
but like Picasso the same way, right? It doesn't. None of these things. It's weird. A lot of them are really like abstract art. And you look
yeah,
at
it's
a
very abstract.
5-year-old could do this, and then others are like, "Holy smokes, this is a talented artist!" Like,
No, it's
oh
like
God, talented.
it's
Yeah,
like it's like the guide says. Like you might say that you can do this because in essence, some of them are very simple, but um, you didn't do it and you
yeah,
wouldn't do it. That's the difference. And
yeah.
he did, and it was his life, and he obviously believed fundamentally in what he did, and obviously he was around the time modernism and Dadaism, so those influences come out and all that. But I love the story. There's one work in particular, and you look at it, it's huge canvas, and it's like a black wavy line going from bottom left hand corner to like say top somewhere short of the top right hand corner. It finishes with a curl or vice versa. I can't quite remember the structure, but that's the gist of it.
Hmm.
But it's like where the guide's saying, well, he would do this and he'd maybe paint a little bit and he'd see where the brush took him and he'd stop. And then he'd stop and think about it for a few hours or a few days before picking up the brush and going that again. I mean, I wouldn't have the patience to do that.
Oh, it's
In hell, I could do that one.
and
But
then well, John, when you look at the art, you can see where he stopped. It's like, oh my goodness, this thing is not it's not just one line.
yeah, sure,
You can
absolutely.
really
So,
see this. What one of them took over a year, I think she said. It was
yeah, yeah. So
that
yeah,
amount of, I don't
it's
have that patience, John.
so when you get the tour guide, it's fascinating to get an insight into the mind of the artist because that mind functions so differently from how my mind functions. And I might sort of like tut and go, "What? Why would he do that?" But it's still fascinating. It was good, really good.
Yeah, yeah, and just to round out, I don't have much to add other than I'm going to switch topics a little bit. The panel I did for Bioanalysis Zone, I should give them a shout out. It was a great panel on oligos, and you can check that out if you're interested. But we went out to dinner afterwards at a place called the Snail. Obviously, whatever the Spanish word for snail is, like escapes me at this time. But that was a really eclectic meal, John. I don't know if you had any other meals, but that was a. This place is like it seemed like a museum. You kind of had to walk through the kitchen, like you got on this old cobblestone road, and you were like really in the heart of Barcelona. And you kind of when you walk in, you're like, "What? This place is tiny." You're walking through the kitchen, and then it's just massive, John. It's 3 stories. This prosciutto hanging from the Iberian ham is hanging from the ceiling. Literally, that is what you're going to eat at some point. And if you get on, I could share some pictures here, but it was one of the best meals I had had, not just because of the great white Spanish wine, but monkey fish is native to Spain, and if you're ever over there, eat the monkey fish. It was almost like a scallopy, buttery, delicious tasting. And then we actually had snails. You can't go there and not have snails, and those were amazing. And then obviously you can't go wrong with some of the Iberian ham. But that rounds it out for me, John. We're right up on the hour here, but it was. And you said it best, John. It was a whirlwind tour, right? I was gone for 5 days, 2 of them traveling, only like 75 hours on the ground in Barcelona. But for me, it was a day of conference, panel at night, that exquisite dinner. Got up the next day of conference, presenting at that, and then we went out to the museum that night, and then I got on a plane and went home. It was awesome, John. It was really really cool. If I do go back to EBF, and I hope to, it's just so close to Thanksgiving. I'm going to try to find a way to go beforehand, John, because I'm
Sure.
tired of going over. I'm tired of going. This is my third trip to Europe this year, and literally, I don't think I've spent a dozen days in Europe in those 3 trips. I'm too old for that, but I'm getting better at it, John. I'll give you the last word, and then maybe we can talk a little bit
Sure.
about what we're going to be up to.
So the restaurant is Los Caracoles. That's what the
It is
name
amazing, John. So you've been there. It is so
I've been
good.
there because I did the panel last year. So they obviously take the speakers to the same restaurants. I did go to a place called Fistmüller, which I've been to before and is good. I would say a couple of starts we had were excellent. A couple other not so much. I was lucky in that because my wife also wanted to go back to the UK. I actually came in and out through Edinburgh this time, so I spent a couple of days in Edinburgh, a couple of days in Fife with my wife's family, then flew to Barcelona, did the whirlwind of the Barcelona meet, and then did a couple more days in Edinburgh. Then did a couple more days in Edinburgh. The problem with Edinburgh the weekend, this last weekend, was I was supposed to be meeting friends for drinks, but I woke up at whatever time in the morning, went outside the hotel, and it was snowing in November in Edinburgh, which I've never seen, and it basically ground all the trains to a halt. So my high school reunion didn't happen. So had a quieter Saturday than expected, but still really nice. Edinburgh is the more I go to it, the more I enjoy it as a city. But yeah, I've had a excellent sort of, I think around 11 days I did total, but it was great. So yeah.
No, that's something, John.
Like
Yeah,
I said,
that's good.
all in all, EBF, the Open Symposium this year was it was a good one, and I had an absolute blast, and I'm looking forward to doing it again.
Well, John, big week here. At least for me personally, I find Thanksgiving to be maybe my favorite holiday for a variety of reasons. Everybody likes maybe Christmas and other things, but this one is fantastic because I don't have to do squat diddly squadoosh other than get the Christmas lights up, which is done. I'm almost done with that. So I'm going to have a nice, we get Thursday and Friday off. I don't think I'm going to take any additional time. But, got my son home from college here, and he came home last night, and that's been fantastic. We'll spend a whole bunch of time with him in the coming days, and then my some in-laws will come over, and then we might have some people over a little bit for dessert and so forth. We make a turkey. We don't brine it, John, because we like it dry. Don't ask me why. We like to pour gravy on the turkey instead of just making it moist, but we do. It's just a tradition, and so. I don't have any good stories about my uni pizza oven or my big green egg, other than one of the panelists shared with him the big green egg, and he found it, and you can buy them in Switzerland apparently, and maybe not quite as big as the one I had, but I did pique his interest in that. So hopefully that was my kind of contribution to dinner talk, is all about my sous vide and cooking, and had a great conversation about making a meat sauce. I got a new recipe from a true Italian on how to make meat sauce. He takes hours to make his, but I'm rambling on and on, John. I'm super excited for Thanksgiving. It's just such a great time. We get to relax for at least Thursday. There's nothing open really, and we relax. The wife and mother-in-law, they like to go out. They used to go out around everything. Black Friday would start earlier. Now Black Friday is going on and on, but sometimes they'll break away around 10 or 11 o'clock at night on Thursday, or they'll try and get up early Friday, but one or the other or both. But they'll be out there shop until you drop. It's not something I do too much of. I just kind of go and maybe pick up packages or kind of relieve them or bring them food, but they'll be shopping all day on Friday, where all I'm going to do is watch football, might work out, and just relax, John. It's one of my, like I said, it's just not as stressful as other holidays because I don't cook. That someone else will cook, which is a relief for me. And then I just literally just hang out, man. Love it. Thanksgiving is great. Football will be coming on. All sorts of good stuff. What say you, John?
So for us, obviously I'm not American, not been raised with Thanksgiving at all, but cannot resist the Thursday and Friday off. And actually this year we thought we'd have a number of the kids coming to stay. Turns out it's just my wife and I, which we're both actually really excited.
Yeah, you need the rest. Yeah.
Because we have a really nice relaxed time. So I'm in the hotel room now. Once we finish this recording, I will jump in the car, drive home, and catch up my wife, pack up unpack things. So I won't be doing any Black Friday shopping because I did it all in Edinburgh. So like make sure that my whiskey got back safe. So the whiskey this time. Again, another thing, the whiskey shops are fascinating because there's all these distilleries I've never heard of. But one of my friends from school, since retiring, works as a guide at Glengoyne Distillery, which is the southernmost Highland distillery. So I just thought, rather than me trying to figure out what whiskey to get, I sent him a text and said, which Glengoyne would you recommend? So he gave me a far more in-depth response than I was expecting. But I got my Glengoyne.
You got a thesaurus, did you? Right? Yeah.
Yeah, I got my Glengoyne whiskey in, so I know it's safe and intact, and that will go home. And yeah, so for us, it's just give me a relaxing time. I don't know if we'll even cook anything on Thursday. I think it would just, like I say, just enjoy the day to just sort of chill and catch up.
Yeah, no, there's nothing wrong with that, John, and
Exactly.
it's such a late Thanksgiving too. It's the latest possible Thanksgiving we could have, so it's like Christmas will come bombing at us this year. That's what's going to happen. But been a fun podcast, John. It was great to see you, and we got to hang out quite a bit.
Sure.
I feel bad for you there. I was blessed to have you around. I'm probably a pain in the butt, but anyhow, it was a lot of fun. Actually, we didn't even talk about our Tuesday night dinner where you and I had it was a decent dinner. I
like I said, I had a great time. No complaints at all.
I didn't realize there were 4 types of I had an Iberian ham, Iberian chorizo, Iberian sausage, and Iberian loin. It was hard to choose which one I like, but the Iberian ham it's near and dear to me. I think if I die, I want to have a big friggin' Iberian ham in my hands when you bury me, because I love it.
Just make sure it's nice and warmed up, so then it's like this slices come off really nice.
Yeah, that's it. It's it. Yeah. It's so and prosciutto's okay, but the Iberian ham is just everywhere there was delicious, but at the Snail it was really good. Even the plate I got at that little restaurant that we were at around the corner from the hotel, which was really good quality.
Sure. Yeah.
But anyhow, John, I think that's it. I'll have a great Thanksgiving.
You
audience listening. See you in the funny pages.
Yep. Thanks very much.